Hi guys, this is Alicia with Good Morning Sunshine. So today we had our bovine lab where we stuck our arms up a cow's butt. So this was the lab, again, I was not looking forward to since I got accepted into vet school. Like, this is the lab I did not want to do ever. But anyways, it was interesting. Would I do it again if I had to? Yes, because vet school. But if I had the choice, I would not do it. It wasn't as bad as I thought it'd be, but it was still pretty bad. The cow pooped like 5 times between me and my friend Zach. Yeah. So not a good time. But we didn't get like poop totally on, so that was a plus. But it was raining and muddy and there was urine everywhere and poop everywhere and it was just not a good time. Not where I wanted to be, but here we are. We made it through the lab. So, we had nutrition this morning. I didn't go to the lecture, but I have the notes, so I'm going to try to go through that with you. And then, of course, 40 minutes after the lab let out, we had lecture to get back to, which we rarely got to. At least we had like 15 minutes to spare, but we had 2 lectures still to do after that lab. So we just took our coveralls off, threw them in a bag, and went back to school for lecture. So, and then we had 2 immunology lectures, so I'll go over those with you too. So starting with nutrition, lecture number 7. It was about microbial contribution. We have a few terminology we have to go over. So CHO means carbohydrates. So if I say CHO, that means carbs. And then we have the gut/GIT, which is the gastrointestinal tract. I think I've told you that one before. And then we have foregut, which includes the esophagus and the stomach, aka it's pre-gastric. It's cranial to the true glandular stomach, and it's cranial to the abomasum and ruminants. And then we have the hindgut, which is includes the cecum and the large intestines, aka it's caudal. And then we have microbes, which equals microorganisms, and then we have microbiota, which equals microbial population with an environment. So gut microbiota. And then we have the microbiome, which equals DNA of the microbial population. So just to give you more clear stuff, here is the picture of a bovine. So we have the esophagus there. We have the big O rumen, which is their stomach, and then we have the small intestine and large intestine here. The abomasum sits here, and the omasum is the round part there. And then we have the reticulum, which is basically the second part of the stomach because food goes in the rumen first, and then to the reticulum, and then back, and then to the omasum to the abomasum, which is the last part. So we have foregut fermenters. And then we also have hindgut fermenters. Hindgut include the horse, the rabbit, rodents, pig, dog, cat, and human. And then the foregut includes the non and modified ruminants. So what kind of microbes live in our gut? We have obligate anaerobes and facultative anaerobes. So for prokaryotes, we have bacteria and archaea, and then for eukaryotes, we have protozoa and fungi. So the gastrointestinal tract is inoculated at birth. So where do the microbes come from then? Well, the health and gut microbiota of the dam influences the offspring health. So what does the host provide to the microbes? Well, like making beer as an example of a fermented product, it's to provide food and feed, meaning the substrate from the microbes, directly foregut fermenters or indirectly for hindguts. We have mastication, which is the physical substrate processing. We have the fermentation chamber, which is the rumen, the cecum, and the colon. And then we have eructation, flactus, and defecation, which is the removal of our the waste products. So then we have volatile fatty acids, which is VFA, production along the gastrointestinal tract of a hindgut fermenter. So the highest concentrations of the VFAs are found in the cecum and the large colon, so for hindguts, and then which coincides with where the majority of the gut microbes are. So that makes sense for hindguts. So we have the fate of microbial volatile fatty acids. So for acetate, the primary metabolic product or role is for energy and fat. For propionate, the primary metabolic product is glucose and lactose. And then for butyrate, the primary metabolic product is enterocyte fuel. So forage diets yields acetate, which yields milk fat, whereas grain diets yield propionate, which yields milk lactose, which equals for milk production. So what percent of daily energy comes from products of the microbial fermentation, so the VFAs? For the cattle, up to 80% can be used for the daily energy from the microbes. In a horse, it's up to 50%. For pig, it's about 20%. For humans, about 10%. And for dogs and cats, is about 5%. So we have microbial protein in the rumen. So I'll show you this. Here's this. So again, changing gears from carbs and energy to protein. Nitrogen enters the rumen as an amino acid nitrogen. From dietary protein, forage, the soybean meal, so anything that we feed the ruminant. And then 2, it goes to a non-protein nitrogen, which is an NPN, which is an example would be urea. And then 3, we have nitrogen in saliva. So the rumen microbes can utilize the nitrogen from all 3 sources to make microbial protein. This is unique in animals' need because animals need amino acid nitrogen and cannot utilize urea without the microbes in the monogastric animals. Monogastric animals meaning the humans, the dog, the cat, the horses cannot utilize urea and must have an amino acid nitrogen. So the rumen microbes need nitrogen from one of these 3 sources plus a carbon source for carbohydrates. Some dietary protein escapes the rumen, aka bypass the protein. The protein that is presented to the ruminants, the small intestine include microbial protein plus the bypass protein. So what about microbial protein hindgut fermenters? Because it's excreted in the feces. So that's what it does. So hindgut don't use it as a protein source, whereas the foregut uses it as a protein source. So then we have dietary fat and microbial lipolysis. As with dietary protein, rumen microbes get access to the dietary lipid before the host does. As compared to the monogastrics, where protein and lipids are digested and absorbed in the stomach and small intestine by the host digestive enzymes. Rumen microbes modify lipids in 3 ways: through hydrolysis, which is the breakdown of triglycerides into fatty acids and glycerol, and 2, hydrogenation, which add double bonds to fatty acids, and 3, conjugation from organic chemistry, alter orientation of 2 double bonds in a polyunsaturated fatty acid. So lipids entering the small intestines of ruminants are typically modified from the form as found in the diet. An example being dietary linoleic acid, which is an omega-6 found in grain like corn, is modified to be conjugated to linoleic acid by microbes. And then we got into dietary fat and microbes. Fatty acids coat fiber particles. So maximum dietary fat for foregut fermenters is about 8% fat for dry matter. And for hindgut fermenters is about 15% fat for dry matter. Why can you feed more fat to a hindgut fermenter? Well, dietary fat coats the fibers and makes them inaccessible to microbes. So what else do gut microbes contribute to? So for vitamins, for that's B12, biotin, and vitamin K. And can we negatively alter the gut microbial population? Yes, we can do this by a diet change or by if the animal is anorexic. So dietary non-structural carbohydrates such as sugar, starch, fructan, and microbes. We have the death of a G bacteria, which yields the release of LPase, which is an endotoxin. The proliferation of G plus bacteria yields vasoactive amines. Leads to mucosal damage. Increased production of the thiamase producing bacteria yields thiamine deficiency. And then we get into why is starch a problem when it should be digested and absorbed in the small intestines? And then can we positively alter the gut microbial population? So yes, with prebiotics, fecal enemas, and transvenation. Also by probiotics, a probiotic is a beneficial microorganism administered in the form of a nutraceutical or supplement administered by nasogastric or orogastric or a rectal tube, and occasionally is put into food. A prebiotic is fiber in food or a nutraceutical supplement thought to increase concentrations of beneficial microorganisms in the GI tract. So again, here's a microbial contribution overview in this table. So function, ability to digest and extract energy from fiber. So foregut, yes, and hindgut, yes. Ability to utilize dietary sugars directly. Foregut fermenters, no. They yield VFA, but for hindgut, yes, they can utilize dietary sugars directly. The ability to utilize microbial protein, foregut fermenters, that's yes. Hindgut, that's no. And then ability to adapt to high dietary fat. For foregut fermenters, that's a no, but for hindgut, that's a yes. So that was the microbial contribution from the nutrition lecture from this morning. And then I will be getting into immunology, but we talked about the innate immunity. So we talked about the first line of defense in the last lecture. So that was, I believe, yesterday or the day before. But I talked about that one. So then now we have the second line of defense, which includes the cellular barrier, which includes granulocytes, which granulocytes include neutrophil, which also can be called PMN, which you probably heard me say in the last lecture. I just didn't explain what it was, but PMN is a neutrophil. And then granulocytes also include eosinophil and basophil. And then the second line of defense also includes macrophages, dendritic cells, and natural killer cells. So we're going to start with granulocytes, so phagocytic cells. We have the myeloid, which include the basophils, the eosinophils, and the neutrophils. So the basophils, they're actually named these different names depending on what type of dye when we're dyeing them and what they uptake. So the basophils suck up the basic dye, the eosinophils suck up the acidic dye, and then the neutrophils, it doesn't take up either of those. But the most predominant white blood cell is the neutrophil. So they come in first, the eosinophils come in second, and then the basophils are third. So we're going to start with neutrophils. They have rapid phagocytosis, but cannot phagocytize repeatedly. So they have little energy and can only kill a few before actually dying. Can only kill a few antigens before actually dying. It has granules, which contain bactericidal enzymes, and they are short-lived. So like I said, they only kill a few antigens before they actually end up dying because they only live a few hours or a few days, and they don't have the ability to present antigen. And then we have the macrophage, which is also called a monocyte. It is slow but can phagocytize repeatedly. It has lots of energy. It can kill and kill and kill and keep killing again and again. It contains the bactericidal enzymes. It's long-lived. And then selected cells have the ability to present antigens. So antigens present to T and B cells. And then we have granules in neutrophils, include lysosomes, myeloperoxidase, superoxide dismutase. And then we have lactoferrin, which binds iron. Therefore, iron is not available for bacterial growth. And then we have collagenase, which dissolves collagen when entering tissues that are invading the antigen. And then we have deficins, which inserts between lipid bilayers and disrupt the interactions of lipid molecules. And then we have a few definitions. We have neutrophilia, which is the increase in number of neutrophils, which signals acute infection is going on. And then we have neutropenia, which is the decrease in number of neutrophils, which signals a problem in the bone marrow, or the pathogen has put out a toxin that is killing the neutrophils. So killing by neutrophils, how antigens are killed by neutrophils. So we have phagocytosis, which includes 4 stages. That's the chemotaxis, the attachments, the phagocytosis, and the destruction finally. And then we have also respiratory bursts, and then we have extracellular traps, which are known as nets. So we're going to start with the first stage of phagocytosis. We have chemotaxis, which is chemical signals that the body sends out as SOSs that attract the neutrophils to the site of inflammation. So we have the release of the chemical signals from the damaged tissue. So this will send out chemical signals that are released from the site of injury or inflammation, which attract the phagocytic which attracts the phagocyte to the site of the pathogen tissue destruction. So then they would adhere to the vascular endothelial cells, and then the increased expression of adhesion on the surface of the endothelial cells, favoring attachment to the surface molecules of the neutrophils. So increase in space between cells of the endothelium. The neutrophils are trying to squeeze their way through the endothelial cell wall in order to get to the site of injury. So diapedesis is when the neutrophils migrate out of the blood vessel and enter the injured site. So we have identification and attachment. These are neutrophils. They have receptors that allow for the attachment of the pathogen. So the phagocyte physically attach to the pathogen. So the second phase is attachment. So neutrophils and antigen attach to trigger ingestion. So we have one, which is the zeta potential, which is both cell surface of the phagocytes and antigens are negatively charged, therefore repel. The zeta potential needs to be reduced, and to do that they use opsonins, which attach to the antigen in order to increase phagocytosis and its efficiency. Opsonins are proteins that bind to the antigen to facilitate phagocytosis and decrease the zeta potential. This process is called opsonization. And then factors affecting phagocytosis, which again is the opsonin. So it's any substances which promotes phagocytosis. This includes antibodies, complement components, and certain liver proteins. The opsonins are the natural ketchup. They're proteins which coat the antigen to facilitate phagocytosis. So then the third stage we have ingestion, and then the fourth stage we have the destruction, which is a fusion of the phagosome with the lysosome. The phagolysosome results in dumping of noxious antigen, which is destroying chemicals. So here is the engulfment of the pathogen, which is the bacteria, and is trapped in the phagosome. So then we have the destruction of pathogen by the lysosomal enzymes. So again, pathogen, bad. We want them out. So it becomes trapped and is bathed in a toxic antimicrobial fluids. And then again, you have the formation of the phagolysosome. So the lysosome granule moves toward the phagosome and fuse to form the phagolysosomes. And then it is ultimately killed. So killing by neutrophils, we have the respiratory burst, which you have that, and then binding to a bacterium results in the activation of a surface enzyme, that being NAD pH oxidase, which yields NADPH. This releases 2 electrons, which releases 2 oxygens. And the oxygens have to go somewhere, so then we have a superoxide ion because the oxygen binds with the 2 electrons in order to become the superoxide ion. And then water comes into play, which yields hydrogen peroxide. And then you have chloride and myeloperoxidase, which make the hypochlorite ion OCl negative. And the hydrogen peroxide and the hypochlorite ion are toxic to the antigen. So you have the killing of the neutrophil. Oh, killing by the neutrophil, sorry. Killing by neutrophils, you also have the extracellular traps, the nets, which are so as neutrophils are dying, they put out this net. So even though they are dying, they are still trying to reduce antigens and kill them off. So they're sticky white web-like nets made of decondensed nuclear DNA. They're coated with antimicrobial proteins from the granules. They are shown to trap and kill bacteria, and they are critical for clearing some infections such as Salmonella and Shigella. Neutrophils form nets and release them as a form of apoptosis, cell death. This is called the netosis. So then we started lecture 4, which talked about innate immunity. So we got into the eosinophil and the basophils. Eosinophil is pink in color and it can phagocytize, but not as efficiently as the neutrophils. It's larger than neutrophils. It's increased in allergy and parasitic infestation. Granules have aryl sulfatase, perioxidase, and acid phosphatase. These dissolve the cuticle of the worm, making it vulnerable to an immune attack. So large crystalloid granule has core of major basic proteins with matrix surrounded by the eosinophil cationic protein, the eosinophil peroxidase, the eosinophil derived neurotoxin, and the eosinophilia, which you assume helminth infestation or allergies. Basophils don't get activated a lot. They're the least numerous granulocyte, so only 0.5% of blood leukocytes. The granules have complex vasoactive molecules. So if you want to see the difference of them, we have the neutrophil, we have the basophil here, and the eosinophil there. So then we got into the second line of defense for macrophages. The anatomic location of macrophage scattered throughout the body. They're called different things. So monoblasts come from the bone marrow, which turn into melanocytes in circulation, and then they move out of circulation, becoming macrophages once they're in the tissue. And then the brain, the lungs, the liver, the kidneys, connective tissues, and the lymphoid tissue, they all call them different things, but they're all macrophages. So in the brain, they're called microglial cells. In the lungs, they're called alveolar macrophage. In the liver, they're called Kupffer cells. In the kidney, they're mesangial cells. In connective tissue, they're histiocytes. And then in the lymphoid tissue, they are known as just macrophages. So then we have resting versus activated macrophages. Resting have a round nucleus, tons of Golgi apparatuses, and mitochondria and lysosomes, and they can produce a lot of different proteins. It is smaller in size, and once it becomes an activated macrophage, it becomes big in size. That's basically what it means here. So you can see the small one, and then the big one is the activated macrophage. So then we have macrophage-derived cytokines. The most important are IL-1, the tumor necrosis factor alpha, and the IL-6. They all induce fever, so that means they elevate body temperature. The IL-6 promotes B cell functions, whereas the tumor necrosis factor alpha induces, induces fever already. Cytotoxic, and the IL-1 stimulates T cells for antibody response. But we have other ones. We have the IL-10 and the IL-12 as well. So food is a gateway for antigens to enter. So when you have a fever, you aren't very hungry because the body is telling you you don't need that food because it doesn't want any more antigens coming into your body. A fever does many things actually. It stimulates hepatocytes, which yields acute phase proteins, which act as opsonins, so again a natural catch-up to enhance the phagocytosis by the neutrophils. It also lowers iron, zinc in blood. This decreased iron will deprive bacteria of iron that is necessary for bacterial proliferation. A fever stimulates fibroblast proliferation, which assists in tissue repair. Fever also leads to muscle proteolysis. This is why we feel achy, which increases amino acids in the blood for the use by the immune system. Fever also decreases the growth of bacteria. Fever also has effects on the immune system, many more. It increases neutrophils in the blood. It increases motility of neutrophils. It increases the alternative pathway of the complement, and it increases the lymphocyte activation, the PT, the BT, and the natural killer cells. This all facilitates in bacterial elimination. So we have the presentation of antigen. So the antigen presenting cells, they are slow responders. They're phagocytosed repeatedly. Their select cells can present the antigen. They present processed antigen to lymphocytes. And then we have the second line of defense. We get into dendritic cells. A dendritic cell is thin members projections. 1, excellent antigen presenting cells, so 100 times more. 2, the antibody-coated antigens are rapidly presented to lymphocytes. There are 4 types of dendritic cells. You have the myeloid dendritic cell, you have the lymphoid dendritic cell, the follicular dendritic cells. These are all hematopoietic origin. But then lastly, we have the plasma cytoid dendritic cell, which looks like a plasma cell. And then dendritic cells are less than 100 times more powerful than macrophages or B cells in the presenting antigens. Dendritic cells are present in all tissues except the brain, eye, and testes. Dendritic cells need a counter with the antigen in order to survive. If there's no interaction with the antigens, they would die in 2 to 4 days. So then we have the second line of defense, the NK cells. So their morphological features are the subset of bone marrow derived lymphocytes distinct from B and T cells. They express CD16, have cytotoxic granules containing special proteins like peripherin and protease known as granzymes. And their functional features include kill microbe infected cells by direct lytic mechanism and kill tumor cells and viral infected cells and secrete IFNG. So that's the end of immunology. So I got all that in today, but I hope you guys all had a good day. Tomorrow's Friday, so you better have a good Friday too.